Mutations in certain types of genes, specifically tumor suppressor genes and proto-oncogenes, may result in uncontrolled cell proliferation or cancer. In a population of purple mountain dragons, throat cancer is a leading cause of death. Mutations in 3 genes, gene X, gene Y, and gene Z, have been implicated in this cancer type. The influence of one of these genes alone does not result in cancer. However, the combination of these genes causes fatal uncontrolled cell proliferation in purple mountain dragons. Gene X has been identified as a tumor suppressor gene. Tumor suppressor genes are genes which code for proteins that identify potential errors in cell growth and prevent the cell from undergoing cell division. Prevention of cell proliferation in a mutated cell can mean either rectifying the mutation or ordering the cell to undergo apoptosis, controlled cell death. If a tumor suppressor gene undergoes a heterozygous mutation, the wild-type non-mutated allele can usually compensate, allowing the gene to continue to function normally. However, a homozygous mutation in a tumor suppressor gene, in this case in gene X, leads to the gene being unable to prevent cell proliferation. The result is a benign mass or tumor in the throat of the purple dragon. While gene X inhibits cell proliferation, making it a tumor suppressor gene, genes Y and Z promote cell proliferation, making them proto-oncogenes. Both alleles code for cell proliferation, so a mutation in one allele that increases cell proliferation is dangerous enough on its own without a second mutation in the other allele, which remains wild-type in a heterozygous mutation. This heterozygous mutation in either gene Y or Z results in the proto-oncogenes becoming oncogenes, which are genes that are constituently or constantly activated, resulting in perpetual cell proliferation. When mutated, proto-oncogene Y becomes oncogene Y, resulting in uninhibited cell proliferation in tissue near the tumor caused by the homozygous mutation in gene X, making the tumor malignant. In the case of the purple dragons, the nearby tissue being affected is that of the esophagus. Once affected, esophagus cells are unable to perform their usual functions and instead are only able to continue to divide, causing degradation of the esophagus tissue. The heterozygous mutation in gene Y combined with a homozygous mutation in gene X results in the malignant tumor. A mutation in gene Y alone would not be sufficient. This is known as a polygenic or multigene mutation. Like gene Y, gene Z is a proto-oncogene, meaning it promotes cell proliferation and becomes an oncogene when a heterozygous mutation occurs. When gene Z undergoes a heterozygous mutation in combination with gene Y's heterozygous mutation and gene X's homozygous mutation, the malignant tumor becomes metastatic. A metastatic tumor is one whose cells have detached from the original tumor site and traveled via the bloodstream to an organ within the body whose cell type is highly similar to those of the original tumor. In the case of purple mountain dragons, the metastatic tumor cells travel from the esophagus to the mouth, causing uncontrolled cell proliferation in a new location, the tongue, and further inhibiting the digestive tissue of the dragons.